I told you, this is kind of out of order. So in your lecture notes, turn back to 44 and 46. So we talked about the ependymal cells yesterday. And remember, the ependymal cells line the central canal of the spinal cord and the ventricular spaces. In certain regions that I'll talk about next, there is an intimate association between the ependymal cells and the capillaries of the brain. And plasma in the capillaries leave the capillaries and pass through the ependymal cells. And the ependymal cells modify that fluid. None of the red blood cells are allowed to get into the CSF, but white blood cells can. And oxygen and glucose, but different concentrations of the ions are modified. And so the plasma is modified to become the CSF. It's the source of the cerebral spinal fluid. And it's constantly being formed, okay, several hundred mils a day. Couple hundred mils a day. And it's being formed all the time. So it has to be reabsorbed back to the blood supply. So it's coming out of the blood, it has to go back into the blood. And this is how it goes back into the blood. All right, as it gets back into the sinuses and is carried back to the veins in the heart along with the venous blood of the brain. So let's look at that cycle of where it's produced. So this structure, I'm going to turn the lights down here, that you see that's red is the source. It's the specialized area where we have capillaries and ependymal cells, capillaries of the pia mater and ependymal cells associated in a structure known as choroid plexus. So we have pia mater and ependymal cells all over, but only in these specialized regions is it increased in amount and called the choroid plexus. When we do the sheep brain, you'll be able to see the choroid plexus. It looks like brown algae, okay? So it starts, the first location that we find it is in the floor of the lateral ventricle. When you look at our ventricular models during lab, this C-shaped structure is the lateral ventricle, and the pink painted material is the choroid plexus. Okay, so when you're looking at this model. So the floor of the lateral ventricle, and here we're just kind of seeing into the lateral ventricle. Most of it's going to be behind the screen because it's in the cerebral hemisphere. It's not on the edge. So we're just seeing an opening into it. And as this forms CSF, under pressure, it flows out of the lateral ventricle through the interventricular foramen into the third ventricle. This weird shape is the third ventricle. We have more choroid plexus in the roof of the third ventricle. That's still making CSF. So under that additional pressure, it flows through the cerebral aqueduct and into the fourth ventricle. And in the fourth ventricle, we have even more choroid plexus. There are several directions that the CSF can flow. It can flow under the cerebellum through the median aperture, or there's two openings on either side, the diamond shape of the fourth ventricle, or it can flow down the central canal and out the bottom of the spinal cord. And then under pressure, it circulates back up and around the brain and passes through these arachnoid villi into the superior sagittal sinus. All right, so this is showing veins, but this is the arachnoid villi. Here's another view. So choroid plexus of the lateral ventricle, choroid plexus of the third ventricle, through the cerebral aqueduct into the fourth ventricle, out the median aperture or the lateral aperture or down the central canal, and then again circulating back up into the superior sagittal sinus. So his mechanism for getting through the superior sagittal sinus, backwards, is through these arachnoid villi, and then actually this one right here. So this is the subarachnoid space, and cerebrospinal fluid enters into the sinus and crosses the arachnoid villus membrane and joins with the venous blood of the veins. And then after that, the flow is exactly the same as we had drew up here on the board for the venous return to the blood. So it's a complete circulation. From the blood in the chorion plexus, back to the blood in the sinuses. Alrighty? So that's where we're going to stop for lecture today. We won't do the spinal cord.